Gaby is constantly being exposed to UV radiation from the sun and harmful pesticides. For now, Gaby cells are going through every step in the cell cycle and dividing equally into two individual healthy cells. If a bad mutation was present in the cell cycle, the cell will alert and cause the activation of p53. A long flexible molecule composed of 4 identical protein chains that are held together by a tetramerization domain at the center. Each chain has a long flexible region that connects to a large DNA binding domain. CHK2 phosphorylates p53 transactivation domain at the serine 20 site, triggering the activation of p21. Once p53 is activated, one of its target genes is the CDKN1A p21 gene. With the help of RNA polymerase, it transcribes G21, which is then translated into the p21 protein. p21 then stops the kinases that are helping the bad mutated cell by attaching to them and desactivating them. The cell then changes its route to the cell repair area where the damage will be repaired, which will stop the proliferation of mutated cells. T53 is a gene that codes for the p53 protein. RNA polymerase helps transcribe the gene, and with the help of other regulatory proteins, they then translate the gene into the p53 protein. Because Gaby is being overly exposed to stressful situations, a bad mutation in the T53 gene will be unnoticed, and RNA polymerase will still transcribe the gene without knowing that it is not correct. In turn, the p53 protein would be missing single amino acids that will eliminate its tumor suppressor function. So, CHK2 kinase will no longer be able to activate it. If it's not activated, it will never help transcribe p21. Therefore, bad mutated cells will be able to pass through the cell cycle without being stopped. A controlled cell cycle can lead to the formation of tumors in Gaby's body. We can all decrease the prevalence of cancer by using a mask, wearing sunscreen, eating a decent diet, exercising, and visiting our doctors for at least once a year.